Hello friends, welcome to my channel. We will start with cellular respiration. Let's start with an overview of cellular respiration followed by glycolysis. Cellular respiration, as the name suggests, it takes place in cell. Now, what is cellular respiration? It is a series of metabolic process in which complex compound is converted into simple compound. In this, energy is also released. This energy is used to produce ATP and some is lost as heat. There are various substrates for this cellular respiration, like carbohydrate, lipids and proteins. Here we will focus on glucose. Example, if one molecule of glucose undergoes complete oxidation, it gives carbon dioxide, water plus 686 kilocalorie of energy is also released. This energy is used in the formation of ATP and some is converted as heat. Now this ATP is used by the cell for various processes. So it is called energy currency of cell. Now, what are the processes that takes place in cellular respiration? First one is glycolysis. It takes place in all the cells. In this, glucose through series of reaction is converted into final product pyruvate. This is partial oxidation of glucose. Now what happens to this pyruvate? Depending upon whether the oxygen is present or not, two types of the respiration takes place in the cell, either aerobic or anaerobic. Aerobic respiration takes place in the presence of molecular oxygen. Anaerobic respiration takes place in the absence of molecular oxygen. This anaerobic respiration we will see later. Now what are the processes that takes place in aerobic respiration? Pyruvate oxidation, then Kreb cycle, and then oxidative phosphorylation, which involves electron transport chain. So we see that glycolysis, it is a unique process which occurs aerobically or anaerobically, both. That means it takes place irrespective of whether the molecular oxygen is present or not. So it takes place in all cells. Now the site. Glycolysis takes place in cytosol, both in prokaryotes and eukaryotes. Pyruvate oxidation, in prokaryotes it takes place in cytosol, and in eukaryotes it takes place in mitochondrial matrix. Kreb cycle, prokaryotes, it takes place in cytosol, and in eukaryotes it takes place in mitochondrial matrix. Oxidative phosphorylation in prokaryotes in plasma membrane, and in eukaryotes in inner mitochondrial membrane. About glycolysis. Glycolysis is made up of two words, glyco meaning glucose and lysis means breakdown. So glycolysis is breakdown of glucose. It is also known as the EMP pathway, that is Embden-Meyerhof-Parnas pathway. It takes place in all cells in the cytosol. So the steps are, first glucose is converted into glucose 6-phosphate. Glucose is 6-carbon compound. The phosphate group is transferred by ATP, which itself is converted into ADP, and the enzyme which catalyzes this reaction is hexokinase. So the first step is the phosphorylation step. So why it is required to convert it into glucose 6-phosphate? We know that glycolysis occurs in the cell. This is the cell nucleus. Glucose enters inside the cell through specific channels, but it can easily escape through the plasma membrane. When the phosphate group is attached, so now the phosphate is negatively charged, and now the glucose cannot pass through this plasma membrane, and this glucose 6-phosphate gets trapped in the cell and the further reaction takes place. In the next step, glucose 6-phosphate is converted into fructose 6-phosphate. It is just the molecular rearrangement in which the aldose is converted into ketose. Glucose 6-phosphate is aldose and fructose 6-phosphate is ketose. The enzyme involved is phosphoglucoisomerase. So the next step is isomerization. Then the fructose 6-phosphate is converted into fructose 1,6-bisphosphate and the enzyme is phosphofructokinase 1. The phosphate group is provided by ATP, which is itself converted into ADP. So this is also the phosphorylation step. Fructose 1,6-bisphosphate is also a 6-carbon compound. Now it is broken down into two 3-carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The enzyme which catalyzes this step is aldolase. So the fourth step is cleavage. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are interconvertible, catalyzed by the enzyme triose phosphate isomerase. This glyceraldehyde 3-phosphate proceeds through the further steps immediately, but dihydroxyacetone phosphate cannot proceed through these steps. First of all, it is converted into glyceraldehyde 3-phosphate and then this goes into the further steps. So now we have two molecules of the glyceraldehyde 3-phosphate, and the fifth step is the isomerization step. This two molecule of the glyceraldehyde 3-phosphate first of all transfers its hydrogen atom to NAD+, which is converted into 2NADH. So NAD+ is reduced and glyceraldehyde 3-phosphate is oxidized. This is followed by the phosphorylation, in which inorganic phosphate is added to this glyceraldehyde 3-phosphate and it is converted into 1,3-bisphosphoglycerate. The enzyme which catalyzes this step is glyceraldehyde 3-phosphate dehydrogenase. So the next step is the oxidation step, and it is the only oxidation reaction in glycolysis. 1,3-bisphosphoglycerate is then converted into 3-phosphoglycerate. One phosphate group is transferred to ADP, which is converted into ATP. and the enzyme that catalyzes this step is phosphoglycerate kinase. This step is the substrate level phosphorylation. Because here the phosphate group is transferred from 1,3-bis phosphoglycerate to ADP to convert into ATP, and this 1,3-bis phosphoglycerate is a substrate. Now in the next step, 3-phosphoglycerate is converted into 2-phosphoglycerate. It is just the rearrangement step. The enzyme involved is phosphoglycerate mutase. From 2-phosphoglycerate, water molecule is released and it is converted into phosphoenol pyruvate, which has high energy enol phosphate linkage. The enzyme that catalyzes the step is enolase. So here the water is released, so it is a dehydration step. In the next step, phosphoenol pyruvate transfers its high energy phosphate group to ADP, which is converted into ATP. The enzyme that catalyzes this step is pyruvate kinase, and we get 2 molecules of pyruvate as final product. This is also substrate level phosphorylation. Because here also the phosphate group is provided to ADP by the substrate PEP. So in this glycolysis, ATP is consumed at 2 places and it is gained at 2 places. 4 ATP molecules are produced and 2 ATP molecules are consumed. We can say the net gain is 2 ATP molecules. 2 molecules of NADH is produced, 2 molecules of water is released. So if we see the overall reaction, glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphate gives 2 pyruvate molecules, 2 NADH, 2 ATP and 2 H2O. Glucose is 6 carbon compound and this pyruvate is 3 carbon compound. So we can say that first 5 steps of the glycolysis where the ATP is consumed is the preparatory phase. And the next 5 steps are payoff phase, in which the ATP is gained. Now, some of the important facts of glycolysis. The enzymes of glycolysis are present in cytosol. So, it takes place in cytosol. It takes place in all cells. It is a unique process that occurs both aerobically and anaerobically. There are 10 steps in glycolysis. 5 steps are known as the preparatory phase and next 5 steps are known as the payoff phase. Among them, the 3 steps are irreversible. And 7 steps are reversible. What does this mean? Here at 3 steps we can see first, third step and the last step. The arrow is showing unidirectional. These are the irreversible steps because these steps are energetically favorable in only one direction. We can convert glucose to glucose 6-phosphate, but if we want to reverse it, conversion of glucose 6-phosphate to glucose, it is not energetically favorable. That means large amount of energy is required, and this reverse step is also not catalyzed by the same enzyme, but it requires some other enzyme. Similar in case of conversion of fructose 1,6-bisphosphate to fructose 6-phosphate and pyruvate to phosphoenol pyruvate. So these are the irreversible steps. Rest steps are reversible. NADH molecule that is obtained, it gives 2.5 ATP molecules in electron transport chain through oxidative phosphorylation. Each molecule of the glucose through the glycolysis gives 2 pyruvate molecule. This is the partial oxidation. It is important to control the rate of metabolic processes that takes place in the cell, because if these go unchecked, then there will be wastage of energy. Let us see the regulation of glycolysis. The rate at which glycolysis takes place is regulated by allosteric regulation of 3 enzymes: hexokinase, phosphofructokinase 1 and pyruvate kinase. Hexokinase, phosphofructokinase 1 and pyruvate kinase. These catalyzes the irreversible reactions of glycolysis. In case of the hexokinase, if the concentration of glucose 6-phosphate is high, it inhibits hexokinase, because now there is no requirement for the further conversion of glucose to glucose 6-phosphate. If the concentration of AMP is high, then these activate enzymes phosphofructokinase 1 and pyruvate kinase, because there is the requirement of ATP production. For this, the glycolysis should continue. But if the concentration of ATP is high, then there is no further requirement of ATP production. So these inhibit the enzymes. Citrate inhibits phosphofructokinase 1 and acetyl coenzyme inhibits pyruvate kinase. We know that pyruvate is converted into acetyl coenzyme A. This then enters the Kreb cycle, and the first product that is formed is citrate. If these are already present, it means there is some alternative source present and the Kreb cycle is taking place and energy is produced. So now there is no more requirement for the pyruvate, and hence citrate inhibits phosphofructokinase 1 and acetyl coenzyme A inhibits pyruvate kinase. Fructose 6-phosphate and fructose 2,6-bisphosphate stimulates or activates phosphofructokinase 1, and fructose 1,6-bisphosphate activates pyruvate kinase. We are going to see pyruvate oxidation. We all know that glucose gives pyruvate in the process of glycolysis. This is partial oxidation of glucose. Now, what is the fate of this pyruvate? If the molecular oxygen is present, we have seen that in cell aerobic respiration takes place and this pyruvate undergoes further oxidation. The site of the pyruvate oxidation is mitochondrial matrix. Site is mitochondrial matrix. Now let us see the reaction. Pyruvate plus coenzyme A gives acetyl coenzyme A. In this process, carboxylic group of pyruvate is released as CO2 and NAD plus acts as the oxidizing agent which oxidizes pyruvate and itself is reduced to NADH. So the 2 carbon of the pyruvate gives the acetyl part of acetyl coenzyme A, and this reaction is catalyzed by enzyme pyruvate dehydrogenase complex. So from this we can say that here carbon dioxide is released and the oxidation takes place. So it is a oxidative decarboxylation process. Second, it is an irreversible process. It is highly exergonic process. That means in this process large amount of energy is released. Now let us see about pyruvate dehydrogenase complex enzyme which catalyzes this reaction. It is an assembly of 3 individual enzymes: pyruvate dehydrogenase denoted as E1, dihydrolipoyl transacetylase denoted as E2, and dihydrolipoyl dehydrogenase denoted as E3. These involve certain coenzymes like thiamine pyrophosphate, lipoic acid, coenzyme A, FAD and NAD+. Now let us see where it is located. This is mitochondria. Outer mitochondrial membrane, inner mitochondrial membrane, intermembrane space, mitochondrial matrix. This is cytosol. So the enzyme is located on inner mitochondrial membrane, E1, E2, E3. Through glycolysis, pyruvate is produced in cytosol and then it enters the mitochondrial matrix, and here the entire reaction takes place. Let us see the entire reaction in detail. Pyruvate dehydrogenase complex is an assembly of 3 individual enzymes: pyruvate dehydrogenase denoted as E1, dihydrolipoyl transacetylase denoted as E2 and dihydrolipoyl dehydrogenase denoted as E3. Coenzymes are thiamine pyrophosphate, lipoic acid, coenzyme A, FAD and NAD+. Pyruvate dehydrogenase along with TPP catalyzes the decarboxylation of pyruvate, CO2 is released and hydroxyethyl thiamine pyrophosphate is formed. Dihydrolipoyl transacetylase then converts hydroxyethyl thiamine pyrophosphate to acetyl lipoamide. This then transfers the acetyl group to coenzyme A and forms acetyl coenzyme A. Lipomide is obtained in reduced form. This reducing equivalent is then transferred to FAD and forms FADH2, that is catalyzed by dihydrolipoyl dehydrogenase, and lipoamide is obtained in oxidized form. FADH2 then transfers its reducing equivalent to NAD plus and it forms NADH plus H plus. This then by electron transport chain through oxidative phosphorylation gives 2.5 ATP. Now this acetyl coenzyme A enters into Kreb cycle. So this reaction is known as link between glycolysis and Kreb cycle. Now what are the functions of these individual enzymes? Here we see that CO2 is released from pyruvate. That means decarboxylation of pyruvate is taking place. Thus pyruvate dehydrogenase catalyzes the decarboxylation of pyruvate. In this step, acetyl group is transferred to coenzyme A to form acetyl coenzyme A. Thus, we can say that dihydrolipoyl transacetylase catalyzes the transfer of acetyl group to coenzyme A. Now, here we can see that the reducing equivalent of lipoamide is transferred to FAD and it is self-oxidized. Thus, dihydrolipoyl dehydrogenase catalyzes the reoxidation of lipoamide. Thus, we can say that pyruvate dehydrogenase catalyzes the decarboxylation of pyruvate. Dihydrolipoyl transacetylase catalyzes the transfer of acetyl group to coenzyme A, and dihydrolipoyl dehydrogenase catalyzes the reoxidation of lipoamide or lipoic acid. We are going to learn about Kreb cycle. Kreb cycle, it was discovered by H.A. Kreb, a biochemist. Hence the name Kreb cycle. It is also known as citric acid cycle. In eukaryotes, it takes place in mitochondrial matrix, and in prokaryotes, in cytosol. Glucose gives pyruvate through glycolysis. Glucose is 6-carbon compound, pyruvate is 3-carbon compound. Now pyruvate undergoes oxidative decarboxylation. It involves the enzyme Pyruvate Dehydrogenase Complex and gives acetyl coenzyme A. This is 2-carbon compound. This then enters the Kreb cycle. In the first step, oxaloacetate, which is 4-carbon compound, reacts with acetyl coenzyme A. This also involves a water molecule. So here the reaction involves aldol condensation followed by hydrolysis and gives citrate, that is a 6-carbon compound. Coenzyme A is released. Citrate plays a central role in Kreb cycle. So Kreb cycle is also known as citric acid cycle. The next step is the isomerization process, in which citrate is converted into isocitrate. This is also 6-carbon compound. This takes place in 2 sub-steps. In the first step, water molecule is released, dehydration. Then it gives cis-aconitate. After that, again water molecule is added, hydration step. Both the steps are catalyzed by enzyme aconitase and gives the product isocitrate. So conversion of citrate to isocitrate is isomerization. Here, molecular rearrangement takes place. Why this isomerization is required? Because isocitrate is better suited for the further steps compared to the citrate. Now in the next step, hydrogen atom is transferred from isocitrate to NAD plus, which gives NADH. This is an oxidation of isocitrate and NAD gets reduced. This is followed by release of one carbon atom in the form of carbon dioxide. So it is an oxidative decarboxylation reaction. This is catalyzed by the enzyme isocitrate dehydrogenase. And the product that is produced is alpha ketoglutarate. This is a 5-carbon compound because one carbon atom is released. Now in the next step, alpha ketoglutarate transfers one hydrogen atom to NAD plus and NAD plus is reduced to NADH. This is oxidation step, followed by the release of carbon atom in the form of carbon dioxide. It involves coenzyme A also. It is also oxidative decarboxylation step and the product obtained is succinyl coenzyme A. This is a 4-carbon compound because here again one carbon atom is lost. Enzyme involved is alpha ketoglutarate dehydrogenase. The product that is obtained, succinyl coenzyme A, it is a high energy compound. It has high energy thioester bond. In the next step, this high energy thioester bond is cleaved. This releases large amount of energy. This energy is then used to transfer phosphate group to GDP, which gives GTP, which is later converted into ATP. Coenzyme A is also released and we get succinate. This is 4-carbon compound. This reaction is called the substrate level phosphorylation and the enzyme that is involved is succinyl coenzyme synthetase. In the next step, FAD removes 2 hydrogen atoms from succinate and it forms FADH2. So succinate is oxidized and FAD is reduced. This is known as the dehydrogenation, that is the removal of the hydrogen atom. And the enzyme is succinate dehydrogenase. This gives fumarate. This is 4-carbon compound. Now in the next step, to the fumarate, one water molecule is added and the reaction is hydration. This is catalyzed by fumarase and it gives malate. Again, from the malate, dehydrogenation reaction takes place. That means hydrogen atom is taken by NAD plus and it is reduced to NADH and malate is oxidized. It gives oxaloacetate. This is a 4-carbon compound. The reaction is dehydrogenation and the enzyme that is involved is malate dehydrogenase. And this completes one turn of the Krebs cycle. Let us understand some of the important reactions of Krebs cycle. One is the isomerization. This is taking place in the step number 2, citrate to isocitrate. Here, both the citrate and isocitrate have the same chemical composition, only the molecular arrangement is changed. This isomerization takes place because compared to the citrate, isocitrate is more suitable for the further few reactions of oxidative decarboxylation. Oxidative decarboxylation. This takes place at two steps: conversion of isocitrate to alpha ketoglutarate and conversion of alpha ketoglutarate to succinyl coenzyme A. Here oxidation takes place. That means hydrogen atom is removed from isocitrate and it is transferred to NAD+. So isocitrate gets oxidized and NAD+ is reduced to NADH. This is followed by decarboxylation. In this, carboxyl group is lost as CO2. Thus, one carbon atom is removed from isocitrate to alpha ketoglutarate also, and from alpha ketoglutarate to succinyl coenzyme A. So 6 carbon compound is converted into 5 carbon compound, and then again 5 carbon compound is converted into 4 carbon compound. So we can say that at the two steps, the oxidation of the substrate is taking place. Next is the substrate level phosphorylation. This is taking place in the conversion of succinyl coenzyme A to succinate. Here the phosphate group is directly transferred by substrate to GDP or ADP, and it is converted into GTP or ATP. Here electron transport chain doesn't take part. So it is known as the substrate level phosphorylation. Large amount of energy that is required is obtained by breaking the cleavage of the high energy thioester bond. Next is the dehydrogenation. This is the removal of hydrogen atom. We can understand this with the example: substrate has the hydrogen atom, and here hydrogen atom is not present. Now this hydrogen atom is removed from this substrate and it gets oxidized. And this is transferred to this certain compound, suppose C. So this is getting reduced. So actually here redox reaction is taking place. One is getting oxidized and the other is getting reduced. In these two cases, from succinate hydrogen atom is removed. So succinate is getting oxidized and it is transferred to FAD, so it gets reduced. Similarly, from malate hydrogen atom is removed. So malate gets oxidized, it is transferred to NAD+ and it gives NADH, so it gets reduced. So on the basis of these reactions, we can tell that oxidation of the substrate is taking place at 4 places. Step number 3, 4, 6th and 8th. That is conversion of isocitrate to alpha ketoglutarate, then alpha ketoglutarate to succinyl coenzyme A, succinate to fumarate and malate to oxaloacetate. Overall reaction of the Kreb cycle. acetyl coenzyme A plus 3NAD plus FAD plus GDP plus 3H2O molecule plus inorganic phosphate gives 2CO2, 3NADH, FADH2, GTP plus H2O. Acetyl coenzyme A is a 2-carbon compound. This 2-carbon is released in the form of 2CO2 molecule. Water molecule is used at 3 places: 1, 2 and 3 places, and it is released at 1 place. We get 3 NADH molecules: 1, 2 and 3, 1 FADH2 and 1 GTP or ATP. So if we talk about energetics, then we are getting 3 NADH molecule, 1 FADH2 molecule and 1 GTP, which is later on converted into ATP. So this is obtained from one Krebs cycle. We know that glucose molecule, that is a 6-carbon compound, it gives the final product pyruvate, that is a 3-carbon compound. So from one glucose molecule we get 2 pyruvate molecules. Therefore 2 Krebs cycles are taking place. So this is multiplied by 2 because it is involving 2 Krebs cycles. Thus we get 6 NADH, 2 FADH2 and 2 GTP or ATP molecule. Regulation of Krebs cycle is important because if it goes unchecked, this will cause wastage of energy and accumulation of ATP and NADH. There are 4 regulatory enzymes: citrate synthase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase and succinate dehydrogenase. Among them, the first 3, citrate synthase, isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase, are controlled by ADP to ATP ratio and NAD plus to NADH ratio. ADP and NAD plus are activators and ATP and NADH are inhibitors. The most important regulatory factor is NAD plus and NADH ratio. Alpha-ketoglutarate dehydrogenase is also inhibited by succinyl coenzyme A, and inhibitor for succinate dehydrogenase enzyme is oxaloacetate. Citric acid cycle intermediates are used in biosynthetic pathways for the synthesis of building blocks like glucose, amino acids, purines and pyrimidines. If these intermediates are used continuously, then their level gets depleted. It is important to maintain certain level of these intermediates so that Krebs cycle goes uninterrupted. For this, there are certain replenishing or filling up reactions present. These are known as the Anaplerotic reaction. The term was given by Kornberg. Anaplerotic reaction by Kornberg. Some of the reactions are: in animals, pyruvate is converted into oxaloacetate. It is catalyzed by the enzyme pyruvate carboxylase. In plants and bacteria, phosphoenol pyruvate is converted into oxaloacetate, catalyzed by the enzyme PEP carboxylase. Similar enzyme PEP carboxykinase also converts PEP into oxaloacetate. Pyruvate is converted into malate by malate dehydrogenase. And then these reactions replenish intermediates like oxaloacetate and malate in the Kreb cycle. So this is all for Kreb cycle. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.